Hi everybody, welcome to the final lecture. So the lecture will be on antimicrobial susceptibility testing, AST. Okay, let's look at the purpose of AST. So one of the most important purpose of AST is to guide the clinician in his choice of suitable antibiotic for patients. So usually, as we learned in the previous lecture, a patient will go through the diagnosis, right? So once you go through the diagnosis, and then the pure culture will be getting from the patient, and then the pure culture will be used to test the AST. And then from the AST, we will know like which drug will kill the bacteria or the pathogen found in the patients. And once you get the information, the clinician will prescribe the suitable antibiotic. Okay, the second objective of AST is to alter an antimicrobial regimen already in use for proper management of patients. As we know, antimicrobial resistance is one of the biggest problems in clinics. So what happens is that when a clinician prescribes an antibiotic, after some time, the bacteria found in the patient become resistant to that antibiotic. So therefore, after doing the AST, we will know whether the pathogen is resistant or not, and then from that information, the clinician will alter the treatment. So which means the patient will be getting different kind of antibiotic. Okay, the third objective is to accumulate epidemiological information on the behavior of microorganisms of public health importance within the community. Again, this comes back to the epidemiology, which means surveillances and the antimicrobial resistance. So from the data that you're getting from AST, usually a responsible agency will collect that information and then they start to study. And then from there, they can pull out the information such as whether the particular community is resistant to particular antibiotic or not. And sometimes from the data, you can also figure out whether a particular community is misusing antibiotic or not. Okay, MIC and MBC. So MIC and MBC are the golden standard in the susceptibility testing. So based on the MIC and MBC, the susceptibility of the particular organism will be determined. Okay, minimum inhibitory concentration or MIC is the lowest concentration of antimicrobial that will inhibit the visible growth of a microorganism. So if it's MIC, the antibiotic just inhibit the growth of the microorganism. On the other hand, minimum bactericidal concentration MBC is the lowest concentration of an antimicrobial that prevent the growth of the organism. So which means it fully kill the bacteria. Okay, there's few methods that you can use to study the AST, and usually this method is recommended by the Clinical and Laboratory Standard Institute (CLSR), and then these four are the most commonly used antibiotic testing, and they are also pretty reproducible. And you have to remember that out of these four, disc diffusion is one of the most commonly and widely used AST in the clinical settings. Okay, the first one is the disc diffusion, and disc diffusion, also known as a Bauer-Kirby method. Okay, and then the standard procedure for rapidly growing aerobic pathogens are well established, especially for Staphylococcus aureus, Enterococcus species, and etc. On the other hand, for the fastidious bacteria, you need to twin the assay a little bit. Okay, especially for Haemophilus species, Neisseria gonorrhea, and Streptococci. And another thing you have to remember that disc diffusion is only provide you qualitative results, it's not giving you any quantitative results. Okay, let's watch the video how a Kirby-Bauer assay can be done.
The Kirby-Bauer assay is a widely used and effective way to test if a bacterium is resistant or sensitive to several antimicrobial compounds simultaneously. You will need the materials listed here to conduct the Kirby-Bauer assay. This next list shows the materials available at each station. To begin, label the outside rim of the petri dish on the Agar side, and label with the organism name, lab section information, and treatment. Next, we're going to use a sterile swab to inoculate the plate. Grab the test tube and remove the cap, and gently flame sterilize. Dip the swab into the bacterial broth. Remove from the tube and gently flame sterilize. Recap the tube. Next, pick up the plate and using the swab in a side-to-side motion, swab the plate gently. The idea is to create a uniform layer of bacteria. To do this, Slightly rotate the plate twice. When the entire surface of the plate has been covered, dispose of the swab in the biohazard container. With a marker, divide the plate into 4 quadrants. Flame sterilize the forceps and make certain the ethanol has burnt off completely and allow it to briefly cool. Pick up a disc carefully and place within 1 quadrant and tap lightly. Flame sterilize the forceps between placing discs. Again, remove the lid, place the disc, and gently tap. Watch this time-lapse video of Serratia marcescens' sensitivity to 5 antimicrobial compounds. Notice the zones of inhibition around each disc. The diameter of the inhibition zone is a measure of sensitivity to a compound, with greater sensitivity forming larger zones and lower sensitivity forming smaller zones. In summary, the Kirby-Bauer assay is a fast and effective way to test potential antimicrobial properties of compounds against a single bacterium. The diameter of the zone of inhibition relates to the sensitivity of a bacterium to a compound, and the assay works by inhibiting growth, not directly killing bacteria.
Okay, the next one is the E-test strip. Okay, it is also known as a gradient diffusion method. And then this method uses a plastic strip with a predefined gradient of antibiotic concentration on one side. And then usually we can get an MIC where the zone of inhibition intersects the E-test strip scale. And then this method is a quantitative method. So I'll explain this in the next slide. Okay, similar to disc diffusion, the E-test strip replaced with drug impregnated disc. So this is our E-test strip will looks like. So if we look at here, so this is the numbers, right? Actually, this is represent the concentration of the drug in microgram per mil. So the concentration from high to lower, and then here you can see a number actually is an abbreviation for the drug. So ME I think is stand for methicillin. Okay, let's see the result interpretation for E-test strip. Okay, this is the E-test strip. Okay, and this gray area is the bacterial lawn. We're showing the bacteria is already grown. Okay, the antibiotic code, and then the clear zones here. Actually, that is the inhibition zone, and then usually the MIC is this part. So which indicating there's no bacterial growth. So let's look at this picture. Okay, so this is the clear zones, right? So this one will become your MIC, and this one there's no inhibition, and this one the MIC will this bit. So its MIC I think is 1.5. Okay, the third one is the broth dilution. So in order to do the broth dilution, so we need a lot of test tubes, broth, as well as agar plates. Okay, so what we first do is we make a two-fold dilution of antibiotic. Now after that, we will add a known concentration of bacteria in all the test tube, and then we incubate overnight. On the next day, we try to find the lowest concentration of the test tube which doesn't allow the bacteria to grow. So for instance, if we look at this one, so this test tube is the lowest concentration that the bacteria didn't grow, right? So this will become your MIC. Okay, this MIC actually is based on the definition here which we learned earlier. And after that, we also transfer these concentration whichever the bacteria doesn't grow into the agar plate. Okay, and then we incubate for another 24 hours. Now after that, the lowest concentration that didn't allow the bacteria to grow will become your MBC, which is this one. So at this concentration, so the bacteria doesn't grow, right? So this will become your MBC. So you have to remember that at MIC, the bacteria is only been inhibited by the drug, but then at the MBC, it's been fully killed. Like for example, Here is inhibited, it grows here. Here it's inhibited, but it grows here. Okay, so therefore you have to remember that if it's MIC, the bacteria only get inhibited by the drug. If it's MAC, is fully killed by the drug. Okay, the last one is the Agar dilution. So Agar dilution is quite similar to broth dilution, but then instead of broth, we will be using a molten agar. So what we first do, so we dilute the antibiotic using a molten agar, and after that we will add it into the plates. Okay, and after that we will put the particular pathogen at the known concentration, and then we incubate overnight, and the next day we can come and count the colonies. So MIC is the lowest concentration of antibiotic that prevented the growth of bacteria. So in this case you can see some colonies, right? And then here are some colonies and no colonies and no colonies. So therefore the lowest concentration of antibiotic that did kill the bacteria will become the MIC. So just a little bit more on the future of antimicrobial susceptibility testing. So again, there's a more gene related technologies coming for antimicrobial susceptibility testing as mentioned here. And then these tests are like, again, it's pretty expensive, but then they're very high sensitive and they're very specific, and then you can get the results very fast. And then sometimes it also works based on the resistant genes, where you can detect the resistance in the bacteria easily. Okay, thank you for your attention. So if you have any question, please post in the e-learning. And one more thing, this topic will not be covered in your final exam. This topic is just to give you a brief introduction on the antimicrobial susceptibility testing. So just keep that in mind. Thank you.